Yeah, so good morning everyone once more. So today I'm going to present the, so this is the title, NGS-enabled transcriptomic insight into plant-viroid interactions and discovery of viroid and viroid-like species. So I was thinking what to present, and then I decided that maybe the best would be to go with examples from our laboratory. So in the first part of the talk, it's not working. Yeah. In the first part of the talk, I will explain you how we discovered this. Yes.
And this.
Mm-hmm. Oh, okay, it's working now. Thanks. Yeah. Okay.
Supposed to place.
Okay. Yeah. So how we discovered the CBCVD in hops with the help of the NGS. And in the second part, I will explain you a little bit of transcriptomics and also show you two examples how interactions can be studied between hop, plant, and viroid using transcriptomic approaches on microRNA level and on the mRNA level. So just a little bit about the hops. Sebastian already explained. So hop is dioecious plant. That means males and females are separated. We are only interested in female plants. So male plants are unwanted. They are eradicated around the fields because the seeded hop cones are not considered marketable, and they are only used in the breeding programs. So, and the female plants are grown because of these cones. So these are ripe inflorescence, and inside there are these chemical factories or lupulin glands which are producing raisins for the beer. So the beer industry actually is 99% consumer of the worldwide hop production. So in Slovenia, the hop is grown on approximately 1,600 hectares. And Slovenian varieties represent more than 90% of the varieties which are grown in Slovenia. So therefore, there is a big breeding program at the Slovenian Institute of Hop Research and Brewing. And every hop growing country actually is devoting a lot of efforts to breed new local varieties. So for example, in Germany, in Hill, there is a breeding institute, in Czech Republic, in Žatec, and also in other countries, they have their own. In US, in Australia, New Zealand, each country has its own breeding program. So how everything started, in approximately 2007, one farmer called Sebastian and complained about the stunted plants in the field he discovered. So the symptoms resembled, so the plants didn't go up to the trellises, they couldn't reach the up of the trellises. And the first thing was that maybe there is a problem with physiology, because some symptoms resembled also that maybe there could be nutrition problems. And then also they tried to find any fungal or viral cause of disease, but they were not successful. So these are characteristics, appearance of the disease plants, and here you can see, for example, vine cracking is really very characteristic for the citrus bar cracking viroid. But at that time we didn't know. And the plants will eventually, in 3 years, they will die. And when you look at the root system, you can see that there is the roots are totally rotted. Then they started to monitor this field, and so these dots means plants with the symptoms, and they followed the field in the following years, and you can see that these dots are getting the frequency getting higher and higher, but you can see the pattern of the spreading is, let's say, in the rows. So that means that there is some kind of mechanical transmission of this phenomenon, and with a very high increment of these stunted plants. Then they started also to report of these plants outside of the original farm. So this was original farm. But when Sebastian was talking to the farmers, they were able to track the source to the original farm. So the farmers are exchanging material, and of course, with exchanging of material, this viroid start to spread around. I already mentioned what they did. They tried some, let's say, common, or let's say these phytopathological approaches. They look under the microscope. They try to grow cut of the stems on the petri dishes plates to see if there are some fungus. Also, they applied more molecular approaches. For example, some primers which could amplify viruses or phytoplasmas, even viroids, but they were not successful. Something new has to be invented, or a different approach has to be used. And at that time, so this was in the, let's say, 2010, 2011, the first papers were coming out about metagenomics approach of the analysis of the disease samples using next generation sequencing. So what is metagenomics? Metagenomics is actually approach where you sequence everything what is in the sample. So like metagenomics of water, metagenomics of, let's say, soil, metagenomics of phyllosphere, when you sequence what's on the leaf. And in this case, we did metagenomics of disease plants. So we were actually encouraged by the publications which appeared at that time that this could be right approach. For example, one of the first publication is that one from the medical field, where the person traveled to the Balkan region, and after a few months he died, but he was of unknown etiology of the disease. But he was a donor of the organs, and they transplanted several organs to recipients. And after a few months, unfortunately, all of them died with the very similar symptoms he had. So at that time, they then sequenced the recipient's fluids, and they discovered that actually he was infected with the arenavirus in this area where he was traveling. So this was kind of the first publication, but there are also a lot of publications were later coming out of from the plant metagenomics studies, especially in grape wines. They were sequencing so-called virome of the grape wines, or you can see here Syrah decline symptoms. So the Syrah cultivar was suffering from the plants were dying soon after the vineyard was planted, and they discovered, for example, which viruses could be responsible for that. So we were encouraged with these publications, and also the symptoms, actually. This stunted growth resembles the HCVD. You have heard Professor Sano, probably Professor Sano's talk, where he talked about HCVD in Japan. So for HCVD, infection is also stunted growth is characteristic. However, those plants are much more stunted than in the case of HCVD. So in the beginning, we thought HCVD. Actually, we found HCVD, but this HCVD present was not actually confirmed in all of the diseased plants. So we were sure that the cause of the disease is not HCVD. And also it was usually present at very low titers, so amplification signal was very low. So because of that, then we decided to go with the NGS experiment. So just shortly about the NGS technology. So NGS is next generation sequencing, and this stands for the technology which came after the Sanger technology. And sometimes you will also heard like next generation sequencing or second generation sequencing, but you can use the term next generation sequencing for all technologies which are appearing. So just briefly, in 2005, the first technology appeared, and this was Roche 454 system, which is using pyrosequencing. What is that? So when the nucleotide is incorporated, pyrophosphate is released, and this pyrophosphate in enzymatic cascade with sulfurase is emitting light. So if the light is emitting, it means nucleotide was incorporated. The system was able to sequence. So what's common for all technologies compared to the Sanger technology? So Sanger technology is sequencing of one sequence per run. For these technologies, so the second term is also high throughput sequencing. That means that you can sequence in one run a lot of sequences. So how much is a lot? 1 million, 100 million, even several billions now. The biggest machine can sequence in one run. So the Roche was able to sequence 1 million sequences. So, for example, the biggest Sanger machine can sequence 96 sequences at one time, and this 1 million. And the reading length was up to 1,000 base pair. The technology was abandoned 5 years ago because it couldn't cope with other technologies anymore, which were able to produce more sequences for less money. After the Roche, Illumina appeared. At that time, the name was Solexa, but then Illumina bought Solexa and abandoned the name Solexa. The technology is actually very similar to Sanger technology, so it's using some terminators to sequence. And for example, they have a lot of different machines. This is the latest one, and you can see, for example, you can get 16 terabytes of the sequences per run. So how much is that? If you divide So this is 16,000 gigabases, so 16,000 divided by 3, which is the size of the human genome. You can get how many human genomes can sequence in one run. So the Illumina currently is covering approximately 90% of the sequencing, let's say, portfolio on the market. Applied Biosystems also came at the same time. This was technology which was using so-called sequencing by ligation. It was able to produce also a lot of sequences, but it was abandoned approximately at the same time as Roche technology. So Thermo Fisher has another technology, torrent technology, which is actually very similar to 454, but instead of measuring the light, so when the nucleotide is incorporated, the H plus is released, and as you know, H plus is related to the pH, so actually it's measuring the change of the pH, and if the pH changes, it means nucleotide was incorporated. But it's a smaller sequencing machine. For example, there were also technologies which were not released. For example, like a helicos, they were advertising it, but it never reached the market. And there are two other technologies which are capable of sequencing single molecule. So for all other technologies, you have to at first amplify DNA to amplify the signal. These two technologies can sequence single molecule. So that means you don't produce any libraries. Pacific Bioscience, so this is the biggest machine, Revivo, and mini-ion nanopore technology, which is, I explained yesterday, sequencing DNA through the pore. They are also so-called long-read technologies because they can sequence much longer fragments than other technologies. So, for example, PacBio can go up to several 10 kbs, and actually Oxford Nanopore is unlimited. You are limited to what you are able to extract, the length of the sequence. So actually they reported that they were able to sequence the fragment in the length of 2.5 million base pairs. So probably with this technology we will be able to sequence the whole chromosomes in the near future. So just briefly, what is required to do the sequencing? So in the beginning, DNA fragmentation, you can DNA is very fragile, so you can share it with mechanical means or with a mechanical, chemical, or enzymatic means. Usually we ligate some adapters at the end. That means we are introducing the known sequences which are then used for either amplification or either in a process of sequencing. And then this library is distributed either in the plates, some kind of small holes, or in case of Illumina technology, these fragments are attached to the surface of the glass and then amplified. So with amplification, you will get arrays of the same amplified sequences, and this will then also amplify the signal, which can be recovered either through emitting the light, or either through, let's say, change of the pH and so on. And then, of course, during the sequences, some signals are generated, which are then converted to A, C, G, and T's. So to go back to our project, we started with collecting material. So previous year we marked all the diseased samples and collected material through the whole year. So for example, you can hear this is beginning of the sprouting, small hop plants, and then they start to climbing, and they climb to the trellises. And we performed RNA isolation. When you isolate RNA, you do the testing. Is it of the good quality? Indicator of these are intact ribosomal bands. And Agilent here, this is the company which invented this terminology, RNA integrity number. So they are using some algorithm to assess those two RNA peaks, and if they are okay, then the RNA integrity number is high. Usually it should be 6 or about to be considered of a good quality. And then we pulled all RNA samples into samples infected or diseased or and healthy samples. Then we performed Illumina sequencing. We performed total RNA sequencing. That means all RNA species were sequenced, and also small RNA sequencing. I didn't mention at that time also publications appeared that during viral infection there is response of RNA interference, and a lot of small RNAs which are actually coming out of the viroids or viruses appear in these samples. We received the results in the FASTQ files. So what are FASTQ files? These are the files where you get the DNA, where you get the sequence and also the quality scores. I mentioned yesterday quality scores. So each nucleotide has a probability, assigned probability, how, let's say, confident we are that this is this nucleotide which is written. And then we performed an analysis. So what we did the de novo assembly of the reads. So that means we used the algorithm to, we were trying to build the contigs. Contigs are contiguous sequences with the evidence that they can stick together, and also mapping to the references. So we took all the viruses from the NCBI gene bank and all the viroids, and these were our references, and we mapped them with the fast algorithms to see if there is a match to the references, and we performed identification. So I will not show you all the results because we don't have much time. Just maybe outcome of sequencing. So we sequence approximately 100 million of the total RNAs, and 12 and 21 million of small RNAs for each sample. Here you can see we did some analysis. So there were a lot of ribosomal RNAs present, what is expected because we did the total RNA sequencing. Then, for example, a lot of chloroplasts, a lot of mitochondria samples, and if even there was a presence of Escherichia coli, we were not sure what happened, but maybe we thought that maybe organic manure was used, and there was probably contamination from Escherichia coli. So this is the results of the small RNA mapping to viroid references. So we took the two samples of small RNA sequencing and mapped them to the viroid references. So in the healthy sample, we got results, or we got hits to, you can see here 1, 2, 3, 4, 5, 6 different viroids. However, when you look at the first one, hop latent viroid, you can see that it was recovered in the full length. it is 256 base pairs. For example, we also get that there is maybe presence of coconut tingaya viroid, but it was only, the length of the mapping was only small fraction of the whole length, so 39 bases compared to 254 length of the whole reference. And also, when you look at the coverage, so what is coverage? Coverage is the term which is explaining how many times each nucleotide in reference is represented by mapping. So average coverage, you can see for Hop latent viroid was 19,000. So that means each nucleotide was represented 90,000 times. So this is really a lot. In other viroids, you can see that this coverage is very, very low. So this is, how to say, maybe drawback of the mapping approach, because those were mapping of really short sequences, and the probability that short sequence will find some hit in any other sequence is quite high. So this is actually some kind of background. And this is expected. All hops which are grown are infected with Hop latent viroid. So, and this is disease sample. Again, you can see hits to a lot of different viroids. Hop latent viroid, it is expected, average coverage 11,000. But here is evidence, so we got the hit to the second viroid. The viroid was recovered and in full length, and it was citrus bark cracking viroid. So already the first analysis give us a hint that there is another viroid present. We did then, of course, also reference mapping of the long RNA sequences, and we got the same results. I'm not presenting. And we also did the de novo assembly of longer RNAs, of shorter, of small RNAs. For example, for small RNAs, we used a special assembly algorithm, which is called PFOR. It is developed that it is actually looking for the sequences which can be assembled in a closed circular form. And again, if you look at the results, you can see for the healthy sample, Hop latent viroid was recovered in the almost the full length, 251. It has 256. And in the disease sample, you can see citrus bark cracking viroid and also Hop latent viroid. Of course, also evidence for some viruses were revealed, which are quite common for hops. So this is the mapping, So this is the mapping of the total RNA to the viroids. For example, Hop latent viroid in healthy sample, coverage 6,000, really high. And here you don't see any background because those sequences were 100 base pair long, so much longer than 21, 22, 24 base pair, and no background. And in diseased, you can see citrus bark cracking viroid and Hop latent viroid with a coverage of 4,000 and 3,000 for both samples. And also de novo assembly confirmed Hop latent viroid in the healthy and citrus bark cracking viroid and Hop latent viroid in the disease sample. So we compiled such a table, and you can see that 4 different approaches: mapping, small RNA, mapping, total RNA, de novo, small RNA, de novo, longer reads. and citrus bar cracking was selected as a very, very probable candidate for the cause of the new disease. For example, here you can see also hop latent virus confirmed with all 4 algorithms, but it was present in both samples, healthy and disease. Hop latent virus is quite common in hops. Hop mosaic virus was not present and healthy, but present in infected, but it was not confirmed in small RNAs samples, so we excluded, and it is known that it's not so strong diseasing hop or important diseasing hop, so therefore we excluded it. So what is citrus bar cracking viroid? We started to investigate. So in the publication from in the first when they discovered it in late 80s or beginning of 90s, I think so. They are claiming that it shows chimeric nature between HLVD and HCVD, if I'm not mistaken. However, when you look at the sequences, you actually don't see this resemblance. The hop sequence had 5 different 5 nucleotide changes compared to the gene-back sequence from citrus. So we believe these are probably also changes which were required that maybe it could jump to the hop. However, only one change, and this is this one, G2T at 272, is probably also changing the structure of the viroid. All other 4 changes are not affecting the viroid secondary structure, at least predicted with the computer program. Also, then we start to analyze the disease plans, and you can see the amplification was highly associated with the disease plans. So these are all the disease plans, and only some of them, they don't have a signal. All of them are, of course, infected with HLVD. And HCVD, as you can see, there is only one small band in plant number 75 is present. So this was, again, good hint that it can be associated with disease. And of course, you have to confirm infectivity. And Professor Jaroslav Matušek from Češke Budějovice helped us with this experiment. So we infected healthy hop plants with a dimeric construct of CBCVD, and we could see the symptoms, stunted growth, and also leaf epinestis, and also we recovered then the CBCVD from these plants. So this was actually the confirmation that CBCVD is cause of this new disease, which we named severe hop stunt disease of hops. So there are just few references which I am suggesting to read, maybe, if you are interested in this field, and I will just take 5 minutes for the second part. Transcriptomics. So definition: what is the transcriptome? So or omics studies first, maybe. So omics technologies are technologies which are studying biological processes on the global level. So the first term coined was genomics. This was early 90s when they discussed the need to sequence the human genome, and they coined the term genomics. So the studying of the whole genomes. And soon after that, the word transcriptomic came. That means all transcripts which are present in the cell. And transcriptomics is the global study which is studying the transcriptomes which are present at a certain time point in the certain tissue, for example. So focus of transcriptomic is which genes are expressed, when, and at what levels. And the goal is to understand functional elements of the genome and cellular responses. So what is the, let's say, benefits of transcriptomics? So transcriptomics capture gene activity rather than just DNA potential. So you can see how the genes are, for example, expressed. And also, it is much, much smaller compared to genome. For example, human genome, I said previously, actually it is 3.2 gigabases, and transcriptome of the human is approximately 20 or 30 million base pairs, so much, much smaller. So you need to investigate much smaller space. It can reveal differences in expression between tissues or conditions. So let's say you compare leaves or the roots, and you can discover which transcripts are active in roots, which in leaves. Then also conditions. For example, you have diseased plant and you have healthy plant. You compare them, and you will see what is the difference. Then with transcriptomics, you can identify biomarkers. For example, some transcript is really highly upregulated. You have seen yesterday in the lecture that some transcripts are, after infection with viroids, can get really high expression, and this can be biomarker for early, for example, early infection stages with the viroids. Regulatory networks and also roles of unknown genes. In the beginning, it was maybe related only to the protein coding genes or to mRNA, but now we can study also the non-coding genes. As you know, there are a lot of non-coding genes. For example, the human genome, as you know, currently we are having less than 20,000 protein coding genes, but we have more than 25,000 non-coding genes, so which are also important and which are also expressed. Methods in transcriptomics which are used. So actually, the first, let's say, study to study the RNA was northern blotting. So hybridization, detection of RNAs. And soon they discovered that actually the intensity of these bands are related to the expression levels. But of course, and also, let's say, the replacement for northern blotting, not complete, is like, not really complete, but similar is RT quantitative PCR, where you can also get hint how some RNAs are expressed. But these are single gene approaches, so you cannot detect all the on a global level. Then in early 90s, Craig Venter did the first mRNA sequencing of human brains. And they showed that actually this is a good technique to find the gene sequences, and also, so this was massive Sanger sequencing of mRNAs, and they were also getting the number of these mRNAs. So if they counted more counts for the same RNA, they got some kind of expression pattern also. And then in the 90s, the first really powerful technique appeared, and this was microarrays. Microarrays are actually miniature northern blots. So you have, let's say, glass slide. On the glass slide, you have all the genes spotted, and you probe them with the probes, and probes are coming from your sample, healthy, diseased, and then you compare the signals, and you get the expression levels. So these were microarrays, and one really big company in this field was Affymetrix. It still exists, although it's much smaller. However, as I said, this was late 90s. In 2005, NGS appeared, and they then discovered that what you can do is just sequence mRNA, count the number of the reads, and you will get expression levels. So the NGS then quickly replaced the microarrays, but they're still used today, and especially in diagnostic field. So you get the number, you count the number of these reads, and you get expression levels. This is just my last slide. So so what is in the cell it represents, and for example, mRNA, so protein coding genes, present really a small fraction. So out of the total RNA, 1 to 5%, let's say, is only mRNAs. Non-coding RNAs are much bigger. You have to bear this in mind. I don't have time to continue with the two examples, so we will share these slides with you, and you can then also, you will see so we performed the RNA-seq experiment between the diseased and between CBCVD infected and healthy plants, and also HLVD infected, and also we were looking at the expression of the microRNAs in the hops with the studying of the transcriptomic analysis. So thank you.